So our buddy Greg, he wasn't satisfied at looking at just one trait. He also started to look at two different traits. So if we cross two traits, so if we take individuals that have seeds that are both yellow and round, and we take another individual that have seeds that are both green and wrinkled, and we cross them together, we would get individuals, we can draw this out with a Punnett square, we will get individuals that are heterozygous for both traits. Okay. Now the question was, what will happen when we make that next generation? So if this hypothesis of linked assortment is supported, we should see 3 round yellow seeds, 1, 2, 3, and 1 green wrinkled seed. So our expected ratio is 3 to 1. Is that what we will observe? We don't know. The alternative hypothesis is if these two traits are independent of each other and they are going to arrange themselves differently during gamete formation, we can do a lot more complicated of a cross. Here we're going to take our F1 generation and we're going to find out what allele combinations are possible for our gametes. Okay, so there's a couple of ways that you can do this. Okay, so we can use this FOIL method, first, outside, inside, last. Okay, so we know that in each gamete, we need to have an allele for the Ys and an allele for the Rs. Okay, and the parent contains these combinations. So in each of the gametes that we are going to make, we need to have a Y spot and an R spot. Okay, so we could link this Y could match with this R. This Y could match with that R. Our little y could match with a big R. Our little y could match with a little r. And these are our gamete possibilities for both the parental line and the maternal line. So those same gametes are both optional. So using this tree method, what you could do is say, here are our possible alleles for seed color, and here are our possible combinations for seed shape. Okay. Then all you have to do is follow this tree, right? So we're going this. It's going to be big Y, big R. This big Y matches with this little r. That's going to be big Y, little r, little y, big R. Oops, little y, big R, little y, little r. Okay. Either method, you're going to get the same combinations. So just choose which one you like that makes more sense for you. So if we now take a look at this cross where we have this F1 generation doing self-fertilization, Gregor Mendel actually found was that he found yellow round seeds 9 times. We had a 9, and then 3 yellow wrinkled seeds, and 3 round green seeds, and 1 wrinkled green seed. And so his experimental results match this pattern, this ratio of 9 to 3 to 3 to 1, thus supporting this hypothesis of independent assortment. And this led to Mendel's law of independent assortment, which says that the alleles with different genes, right? So here we're talking about flower color as an example, and seed roundness, okay? So making sure that we're understanding the differences between alleles right? So purple and white are the alleles for the trait of flower color. Round and wrinkled are the alleles for a seed shape, okay? So here we're talking about the alleles of different genes are going to sort independent of each other during meiosis.